And good afternoon, everyone. I'm Dr. Kim Williams, and we are at Research Grand Rounds. I really appreciate Dr. Michelle Barati, who is an assistant professor in nephrology and hypertension, and she will be giving a discussion of food preservatives. And for a proper introduction, we're going to have our wonderful division chief and vice chair for outreach. That is Dr. Oh my goodness, so good. That's what I get for yeah, just running up the stairs. Dr. Rosemary Usa. Okay, and hopefully she's not as short of breath as I am.
No.
All right. Thank you so much.
Oh, that's good. That's what I asked. Hello, everybody. It's my pleasure to introduce Dr. Michelle Barati. Dr. Barati did all of her training here at the University of Louisville, worked her way up through the ranks. Her focus has been on visualizing and imaging of cells. But to get there, she is also an expert in isolating kidney tubular cells and other cells for them to get examined. She is working with multiple different people, both with people studying lupus, studying other proteomic projects. This is one of her projects, and it's actually fascinating. So I'm sure as you're eating your lunch, you'll look at your lunch and say, oh, do I really want to eat that today? I don't know. So the topic for today is food preservatives and kidney disease, from histology to spatially resolved proteomics analysis. You're going to make them stop eating those potato chips, right?
Yeah, right.
There you go.
Thank you.
Eat them now.
Well, thank you, everyone. Hopefully I'll keep this going at a good rate. All right, so the goals for today, we have a few goals. One is, I'd like to go ahead and discuss the association of food preservatives and kidney disease with you. And then I kind of want to divert after kind of going on with some studies that we started looking at the effect of a commonly used preservative, sodium benzoate, on the kidneys. And then within that, and kind of discussing, what our workflow is to address what's happening with the kidneys in relation to sodium benzoate. I kind of want to go through sort of a historical perspective of how to isolate different structures in the kidney and the different methods that we've used literally since the time I've been with nephrology, and actually before nephrology. Okay, so let's get started. So the NOVA food classification system is basically classifies foods into 4 categories based on the amount of processing. So category 1, and NOVA is really just all it means is new classification. So it's not an acronym or anything like that. It just means new. Food category 1, NOVA food category 1 is basically whole foods, fruits and vegetables, meats, no processing whatsoever. Food category 2 are basic culinary ingredients, butter, flour, oils that have had some level of processing. They're pretty low. Food category 3, NOVA 3, is basically where they've taken NOVA 1 categories and mixed them with NOVA 2 to begin and to make something. So like homemade bread or artisan bread, some processing, for example, to get things like oat milk, sauces, tomato paste, that kind of stuff that you typically use for cooking, and then some canning. And then finally, NOVA category 4 of ultra processed foods. This is where basically things are hardly recognizable. I mean, they have like no nutritional value. They're stripped of all their nutritional value. They're filled with fats and salts and sugar. They're hyper palatable. They have a lot of oils, low in fiber, and a high amount of salt and sugar, like I already said. Sorry. And interestingly enough, there are actually apps that you can use if you're curious to know, what am I eating? Where does it fall within this? So you can literally go to your phone and download these apps. One of them this is literally what they're saying, the NOVA Food Processing Scanner. And then the Nutri-Score scan is really interesting. You can actually scan the barcode on foods, when you're shopping, and you can kind of see where does it fall on the list. And why is this important? Well, the CDC, National Center for Health and Statistics, has, it publishes every now and then on ultra processed food consumptions in, youth and adults. And their latest publication in 2025, looking all the way up to data that they've collected until 2023 is really interesting when you actually see that adults are having around 50% of their caloric intake from ultra-processed foods. And in youth, even more, over 60%. And if you actually look at the composition of what it is that they're eating, it's the stuff we're having now, right? The sandwiches, sweet bakery products, savory snacks, chips, all that good stuff. But this is the composition of it. And obviously, sweetened beverages being a really big one in both youth and adults. And why is this important with regard to kidney disease? Well, more and more the association between ultra-processed food consumption and the risk for chronic kidney disease is becoming apparent. And so this was a study conducted with the ERIC cohort study. So it's the atherosclerotic risk in communities cohort. And they basically had 14,000 individuals, men and women between the ages 45 to 64. They had no CKD. They were followed from around 1987 up to 2018. And so they had them fill basically a food questionnaire. They filled it at baseline visit 1 and then baseline in the visit 3. 5 to 10 years later, the food items were classified according to a NOVA classification system. And it was a questionnaire that they had to fill. And there were reasons, for exclusion. For example, if they were missing more than 10 items on the food frequency questionnaire, missing covariates, implausible energy intakes, if they were on the extremes, if they were either taking too few calories or too many calories, they were excluded. And if they had an underlying low level of kidney disease, they were also excluded as well. So the total daily consumption for each participant was averaged, and it was an adjusted energy using the residual method. The outcomes that they were looking for were basically meeting any one of the following criteria over a total follow-up of 32 years. So either reduced kidney function accompanied by greater than 20% eGFR decline at any follow-up study visit relative to baseline, hospitalization involving CKD stage 3 and higher, diagnosis, death involving CKD stage 3 and higher, and then kidney failure with replacement therapy, either by dialysis or transplantation. So any one of these criteria would meet their primary outcomes. So during the median follow-up for 24 years, there were over 4,800 people that had an incident case of CKD, chronic kidney disease. The incident rate for the quartile 4, so these were the people that were in that higher processed food, the NOVA category 4. So the rate of consumption with them, the incident rate, was 16.5 per thousand person. And for quartile 1, the lowest level of processed food was 14.7. After adjusting for confounders like lifestyle factors, demographic characteristics, health behaviors, the participants in the highest quartile of ultra-processed food consumption had a 24% higher risk of developing chronic kidney disease compared to the lowest quartile. There was approximately a linear relationship between the observed between the ultra-processed food intake and the risk of CKD. By substituting even one serving of ultra-processed foods with minimally processed foods, there was a 6% lower risk of CKD observed. So that's pretty substantial, actually, when you think about that. There was a more recent study as well in the American Journal of Kidney Disease, and in this one, they used the patient cohorts from the CRIC study, the CRIC cohort, and so that's the chronic renal insufficiency in communities cohort. And for this one, there are, in this cohort overall, there are about close to 4,000 adults. They already have reduced glomerular filtration rate or kidney function. They were enrolled between 2003 and 2008. They had 6-month follow-ups where they would go in person one time, the next one would be a telephone call. And they were excluded from the study if they skipped more than 12 items on their diet assessment forms. Again, if they were at the extremes of caloric intake, if they had any missing covariate data for which they really couldn't relate to later on for the study analyses, and then if there were missed follow-up studies. So at the end, they were left with 2,600 for the CKD progression study. And for this, the dietary intake assessed at baseline, year 2, and year 4 of follow-up. Participants, they recorded their portion size, their consumption frequency, foods and beverages during the previous year of their recording. Okay. So primary outcomes with this study, 50% reduction in glomerular filtration rate or kidney function from baseline, or initiation of kidney replacement therapy, so dialysis or transplant. And they used a CKD-IP method for determining GFR. The secondary outcomes were all-cause mortality or incident cardiovascular disease. And this could have been a composite of myocardial infarction, heart failure, or stroke altogether. On average, the median processed food uptake was about 5.6 servings per day in the 25th to 75th percentile. And the big contenders were about, the sweetened beverages, the snacks and the sweets, and processed grains like cereals and what have you. Okay, so looking at the data, if you want to see between breaking down, tertile 1 is basically the lower processed foods, tertile 2 is in the middle range, tertile 3 is in the ultra processed NOVA category 4, and to see if they develop the CKD progression, whether or not it was statistic. There was a 33% higher risk in the group with the higher consumption of processed foods. If they adjusted this for demographics, so sex, age, income level, education level, and what have you, it was still significant. So there was still a 22% increased risk in this community. If they took that model 1, which was the adjusted, and then they also added eGFR and proteinuria to it, at this point, they're beginning to, the differences are no longer statistically significant between the different consumption quartiles. And same thing, if they continue to add on to the model by adding BMR, adjusting it for BMI, diabetes, systolic blood pressure, and blood pressure control medications, again, the significance was lost. All-cause mortality was significant in all categories. So whether it was the crude or the adjusted for demographics, BMI, eGFR, and so on, they all remain statistically significant for the group in the ultra-processed food category consumption. Oddly, for the cardiovascular disease, none of them were significant. It was reaching here in the crude, but all of the adjusted ones, there was no significance between the groups, regardless of the consumption. Now, this is really strange. When they looked at the hazards ratio of group 3, again, the group that has the highest level of processed foods compared to group 1, the tertile 1, and they compared for sex, diabetes, or hypertension, there really wasn't a significant difference for any of these variables. However, if they had CKD stage 1 or 2, they had a higher risk of developing CKD if they had if they were in the higher process consumption group compared to the ones with a higher level of CKD to begin with. And they think that this might be due to the fact that the group in the higher level of CKD, their diet is already different and adjusted. They're taking less protein, they're probably really watching their salt intake and what have you. But this was another interesting finding relating the two together. So if we were to start to think about what the actual contents are within these foods that are being consumed, the ultra processed foods, and if you start to think about the preservatives that are being included, well, it's a laundry list of things that could be included. But one of them that we're interested in are the benzoates, or sodium benzoate. Benzoic acid is naturally occurring. You can find it in berries, fruits, some spices. very prevalent. And basically sodium benzoate is the sodium salt of benzoic acid. So you react benzoic acid with sodium hydroxide, you have sodium benzoate. Sodium benzoate is very commonly used in foods, personal care products, medications, and it extends shelf life. For food preservatives, it's favored because it's highly soluble and it sort of acts as a flavor enhancer for really aesthetic things. And if you just look around your house and start looking at the labels, you're going to see it. So it's always a fun exercise with the students to say, go home or, go to the stores and just start looking and taking pictures, and they're always shocked. When it's metabolized, it's basically converted to benzoic CoA by benzoyl CoA ligase. And then the benzoyl CoA is converted to hippurate by glycine and transferase, and then it's actually excreted from the kidneys as hippurate. So, are there regulations for the use of SV? And there are. The FDA Code of Regulations approved in 1977, and then it was approved again or updated in 1984. Says that it's generally regarded as safe with a usage of 0.1% in food or drugs by weight. Okay. The WHO limits of SV safe to ingest were increased in 2021 from 0 to 5 to 0 to 20 milligrams per kilogram of body weight. It has been actually used in clinical settings for therapeutic purposes with a range of neurocognitive disorders, as well as it's not commonly used, but I think it's last line of therapy, if ever, at least from what I hear in our kidney talks, for use with urea cycle disorders and its ability to conjugate with glycine and help with elimination of ammonia. Does it have adverse effects? Well, in a perfect chemistry situation, if you had a test tube and you added ascorbic acid and metals and heat and what have you, which isn't very commonly found in this storage of foods, it has the potential to be converted to benzene, which is then, in that case, a known carcinogen. It has been shown to affect the release of leptin, and so for appetite suppression, which could lead to potentially, weight issues. And then it has shown to potentially have a role in altering immune response due to its effect on release of histamines and prostaglandins. What about in the kidneys? Well, interestingly enough, early studies trying to look at the effects of SV in the kidney in the 90s, they were actually utilizing it to see if it would attenuate kidney disease in response to really strong nephrotoxic agents. And that's because it can actually scavenge hydroxyl radicals. And so the early studies, they looked at acute kidney injury basically in response to gentamicin, an antibiotic, or they were using puromycin aminonucleoside nephrosis model, which causes proteinuria and nephrotic syndrome by injuring the glomerular podocytes and endothelial cells through generation of reactive oxygen species. And also with rats that were injected anti-megalin antibodies, which bind to the podocytes. In the rats, podocytes actually express megalin. And so this immune complex leads to basically addition of circulating antibodies that also come and deposit within the glomerulus and leads to severe proteinuria. So in all of these injury models, SB was actually shown to attenuate proteinuria and injury. In the doxorubicin model of kidney injury, it was not able to attenuate kidney injury. More recently, though, with the interest in consumption of altered food process, now people are actually researchers are starting to look at the direct effects, the solitary effects of SV. And there are some studies showing that potentially there's an increase in serum creatinine or serum urea, suggestive of kidney injury and filtration, glomerular filtration loss. And they've also found markers of oxidative stress by decreased abundance of superoxide dismutase to break down superoxide radical, decreased catalase abundance, decreased glutathione abundance, and so on, and decreased MDA or marker of lipid peroxidation. It's also, SB can also behave as a D-amino acid oxidase inhibitor, which basically can oxidized D-serine, and D-serine in high doses can actually lead to tubular necrosis, proteinuria, and glucosuria. And in that case, they actually found that it was able to attenuate by inhibiting D-amino acid oxidase, and they were actually able to inhibit the D-serine induced proteinuria in mice. Okay. Now, despite regulations of SB usage, effects of increased consumption of foods containing SB, coupled to a high prevalence of obesity and metabolic syndrome and hepatic steatosis, that's not well defined. So even though the solitaries are being looked at in combination, we don't really know what's going on. And so Dr. Jofflin, who was in the, the GI and hepatology division, she developed an animal model. She was interested in this, looking at liver disease. So she took male and female mice and she put them on either a control diet or high-fat diet. And then they received a gavage of either water or sodium benzoate. And the actual duration was, they were started on the diets, either the control or high diet. And this went on for about 6 weeks, after which, on top of continuing with the diets, they received sodium benzoate for an additional 4 weeks. This is the composition of the control and high-fat diet. And so the percent by weight of fat in the high-fat diet compared to the control diet was 21% compared to 5.2% in the high-fat diet. The sodium benzoate gavage, it looks like it's really high at 99 milligrams per kg. But if you actually consider how much food a mouse would eat during a day, so at around 2 grams of food per day, 2 to 4 grams of food per day, if you were to calculate what 0.1% by weight of sodium benzoate would be, that's literally where it would end up based on that level. So we were really interested in this model because we already know what high-fat diet, what it does to the kidney. It's a risk factor for chronic kidney disease. And in animal models that we have, it's associated with metabolic syndrome, inflammation, lipid accumulation, oxidative stress, mitochondrial dysfunction, and fibrosis. What we don't know, though, is what happens when you combine the two together, sodium benzoate and high-fat diet. And so in these mice that Dr. Jofflin had, body weight in response to high-fat diet was increased in the male mice compared to the control diet mice, but addition of sodium benzoate with the high-fat diet was not additive to the body weight gain in these male mice. The female mice, we did not see significant differences between the groups, except by two-way ANOVA. But the kidney weight all the way across in the males were higher compared to the females. But when we normalize it to the body weight to look at the relative kidney weight, again, because of the increase in body weight in the males, the relative kidney weight was reduced in high-fat diet, and it wasn't any more additive with sodium benzoate. Now looking at the histology. So here's a PAS stained section of kidneys from female mice in the controlled diet. And, as you would expect, the structures, the tubules are back to back. We don't have expansion of the interstitial area. We have nice brush border staining in the proximal tubules. The mesangial matrix looks fairly evenly distributed, and we can see nice open capillary loops. We're not really seeing any accumulation of interstitial cells suggestive of inflammation or interstitial proliferation. If we look at what high-fat diet, for the most part, structures are still pretty back-to-back. The findings, we're not finding any kind of like rampant expansion of the interstitial space. We're not finding drastic changes in the glomeruli as well. We still see that really nice brush border staining of the proximal tubules, and they don't look dilated or degenerative. In SB, we're starting to see some focal areas of interstitial expansion, okay, with a combination of cells that are either proliferating or infiltrating cells and some matrix deposition. But again, these are just like few and far between focal areas. Not, for the most part, some structures are still back to back. This sloughing off appearance is more of an artifact, a fixation artifact, and not an actual active process in these mice. In the combination of high-fat diet and SB, we do start to see some degenerative changes. So we start to see some necrosis and kind of, it looks like literally like the tubules almost being twisted, which is a combination of early necrosis and some degeneration happening. We also have, you know, a tubule that is probably going through either some kind of injury-induced proliferative state, but it is injured. You can see the amount of, let me get my cursor here. The amount of cytoplasm to nuclei is greatly reduced in these, in this tubule. And so again, some focal examples of injury, but not rampant, and we don't have, you know, a severe expansion of the interstitium. In the male mice, again, in the control, about the same. You know, all the structures are nice and back to back, and we have healthy proximal tubules. With high-fat diet, this is, you know, always a process that we see. We see heavy vacuolization, okay? And part of that is lipid accumulation, as well as some degeneration, degenerative changes occurring that we see with changes in cell nuclei. With SB alone, we see that as well. So compared to the female mice, where it was mostly quiet, we actually see this vacuolization occurring, so that we had a different injury response happening in the males. And then in combination of high-fat diet and SB, this is, you know, an example of an area with fibrosis. You can see we don't really have a recognizable structure. It's just basically a lot of matrix proteins and accumulation of nuclei. But again, focal. We don't have it all over the section. And again, continued examples of the vacuolization happening on a regular basis anytime the males are on a high-fat diet. For the glomeruli, there was an undergraduate student who began to analyze, you know, kind of doing some morphometric analysis. So she was actually measuring the surface area of the glomeruli. And then, you know, see this bright red on top of the mesangial matrix, just mesangial matrix, and actually counting the cell number in the nuclei as well. For the glomerular cell number, we didn't find any statistically significant differences between the groups within or between the male and female mouse groups. We have some trends. But with regard to mesangial matrix, we actually see that we're reaching significance in the male high-fat diet. Okay, so the amount of matrix, the mesangial matrix and the glomerulus, which is basically, we have to have matrix, you know, for structural integrity of the glomerulus. But in disease processes, it actually is produced, and it can be produced in excess of normal levels. So we're starting to reach that. In the male high-fat diet plus SB, it looks like it might be reduced, but actually if you look at the glomerular surface area, keep in mind this is the PAS stained area divided by the surface area. So it seems like we're starting to see this trend in glomerular surface area, which could explain why this ratio is somewhat reduced. But it looks like we are actually having an active process happening in the glomeruli in the double treated males. Okay, so given what we know in the literature with looking at markers of oxidative stress, whether in the serum or in some whole kidney tissue, we wanted to know whether or not we're seeing any changes in antioxidative response pathways in whole kidney extracts. So one of the first things we looked at was the enzyme superoxide dismutase, which is critical and one of the first lines of defense in response to oxidative stress and breakdown of superoxide radical. And looking at female and male mice with either control or high-fat diet and the different gavages, we did not find any differences in sodium superoxide dismutase protein abundance. The next thing we looked at was the enzyme glutamyl-cysteine synthase, which is the rate-limiting step in the production of glutathione. And glutathione is heavily required by cells for maintaining redox balance and for proper function of other antioxidative enzymes. Looking at that, here is GCS. In the female mice, we see, even though there's somewhat of a trend, that's not significantly different. However, in the males, compared to control male mice, we see that sodium benzoate alone, high-fat diet alone, and the combination of the two significantly diminishes expression of GCS. So that would suggest that the rate-limiting enzyme production of glutathione is reduced, therefore glutathione production would also be reduced. And the potential for oxidative stress would be really high. So given this knowledge, then we want to know, that was from whole kidney extract, what specific structures in the kidney have that altered abundance? And looking at the kidney, it's not uniform all over. And so when we take that kidney and we grind it up and we do an immunoblot, we have no idea where that's coming from. And you can see the cortex, we have this nice outer renal cortex and a medulla. And if you actually look at the functioning unit of the kidney, which is the nephron, we have the glomerulus, which is the filtering unit that filters blood. And in that filter, it traverses the kidney tubules, for which, depending on which region they are, whether it's a proximal tubule, the loops of Henle going into the distal tubule and the collecting tubule will determine what will be reabsorbed from that filtrate and what will be secreted into that filtrate to ultimately exit the body as urine. And so we want to know, where is that happening? Which part of the kidney is, or the functional unit of the nephron is that happening? And so we have several ways of going about that. On top of that, it would suggest, given the fact that we've seen the changes in ECS, do we have changes in the proteomes of specific And so this is a really simple study that my colleague Tim Cummins and I did recently. And even though it's simple, the goal of it was to basically show that you really don't have to have a lot of fancy tools to begin to separate parts of the kidney and see differences that you normally wouldn't see if you looked at the whole kidney. And so we basically took mice and we had their two kidneys. One kidney we basically sliced up and it was processed, the kidneys were homogenized, and that homogenate was then processed for the proteomic workflow and analysis of the proteome in the whole kidney. The other kidney, we took the slices in between and we actually teased out the cortex from the medulla. And then the medulla, again, just like the whole kidney, was processed for the proteomic workflow and proteomic analysis. The cortex we took, we minced up with a razor blade, and then we digested it with collagenase. And then we ran it over some sieves, sequentially from 100 micrometer to 70 micrometer, to begin to separate out the glomeruli from the tubules. And so what you can see here is we had a suspension of cortical tubules here. And once those were isolated, again, they were processed and proceeded with the proteomic workflow. And the whole goal of this question is, will we see differences? We did this process for normal mice and diabetic mice. And the idea is, what kind of differences can we see if we just do this, literally this gross anatomical separation, as well as comparing it to this tubule suspension? And this is just kind of like a general representation of things that normally you wouldn't see. So this is the whole kidney. If we were to take that whole kidney, we wouldn't see these unique proteins that are being regulated by diabetes within the tubules alone. Those are totally unique and not shown in the whole kidney analysis. Same thing with the medulla. The medulla is very different compared to the cortex with relation to the tubule subtypes that are in there. Okay, so we have the thin portions of the loops of Henle that are playing a really important role in reabsorbing water and concentrating urine. And so this very simple exercise basically showed at a gross anatomical separation, we can reveal changes in the different portions of the kidney in diabetes that would normally be missed if we were to just take the kidney and grind it up. So taking that isolated tubule separation, we've just banked on that protocol that we've had. So we've had multiple papers where we took these tubules, we looked at specific protein pathways. And so here's another paper where we were interested in looking at the ER stress pathway. We found differences in some chaperone proteins by homogenizing these samples. And then we actually took it to the tissue level to see which specific tubule, because keep in mind, this is a tubule suspension of the cortex, so it can include proximal and distal tubule, even though the majority of, 80% of the mass is going to be proximal tubule. And in that, we were actually able to find that, in the diabetic mice, we see a specific induction within the distal tubules compared to the proximal tubules, which are marked with the green and the red here. And then again, my same colleague, Tim Cummins, in his work with David Powell, they took the tubule suspension, they did proteomic analysis, and were able to find very specific changes, as well as a whole network of protein signaling molecules regulated with diabetes. So a very effective tool to, again, specify what structure we're going to look at. That's tubules. We've also done work in glomeruli. This is a previous work very similar workflow to the other one where I showed you the gross dissection. The only difference is before the mice were sacrificed, they were perfused to the heart with small magnetic beads, dynabeads. And so what ends up happening is they stay within the glomerulus. They don't get filtered by the glomerulus. And so when we continue with that mincing of the cortex and then separation over the sieve, what we end up with is a suspension that we can hold up to a magnet. And because the glomeruli contain these little dynabeads in them, they will stick to the magnet and we get much better separation and enrichment of glomeruli. And so this was used quite a while back by our lab. And we were able to, in the old days of 2D gels and proteomic analysis, we were able to find in the glomerular proteome an increase in superoxide dismutase that we were able to validate by immunohistochemistry in the diabetic mice compared to the non-diabetic mice. And interestingly enough, if we took the whole kidney and we homogenized it, we actually saw a decrease in SOD in the diabetic compared to the non-diabetic. And that too was shown in the immunohistochemistry. So we combined tissue isolation with spatial resolution. Okay. This isolate told us that SOD was high. The IHC confirmed it. The whole kidney analysis showed us that SOD was low, and the IHC confirmed it. So SOD, the majority of the cortical area is taken up by tubules, and you can see the expression, the abundance of SOD was reduced in diabetic mice compared to normal. So again, very basic, but effective. Okay, so what do we do when we don't have fresh tissue? A lot of times, we collect tissue, we don't have time to process them. Those processes to separate the tubules and the glomeruli, we have to work with the tissue while it's fresh. Once it's frozen, we can't go through it. Okay, and this is where the laser capture microdissection really comes in handy. Okay, because at this point, we can work with frozen sections, we can work with formalin fixed paraffin embedded sections, and it now gives us the ability to specifically go and cut out and microdissect and capture specific structures. And this process is basically you have a microscope with a stage and a slide on it and a capture device right underneath the slide, literally shown by the simplistic cartoon. So you can take your tissue section, you can find your region of interest, and you would basically use the laser capture dissection microscope to cut out your area of interest, and it literally, in the system that we have, falls by gravity into a collection device. So we've used this method. Mike Merchant in our division worked with a clinical fellow when she was doing a research rotation, basically, well, actually she was doing an extra year of research in nephrology. And so he spearheaded this project with her where they basically went through a kidney section. This is a human kidney section, and they cut out the glomeruli, and here are all the little glomerular sections in the collection cap. They did it in FFPE tissue as well as in frozen tissue, and they basically developed a extracellular matrix proteomic set of database, I should say, from normal human glomeruli. This was then extended, the study was extended to actually look at focal segmental glomerulosclerosis, the fibrotic kidney glomerular problem or disease, I should say, sorry. And in this study, Mike Merchant basically was able to delineate protein abundant differences between two different variants of FSGS. So there was a collapsing variant here, CFSGS, compared to FSGS NOS, or not otherwise specified. And so again, really helps you tease out, and these are from just FFPE kidney sections. So coming all the way back now to our SB study. I had a student, Olivia Osage. She is currently a second year medical student. She worked in the lab this summer. She was really interested in this project. She spent a good amount of time just kind of learning histology and looking at the tubules and the glomeruli and trying to decide what she wanted to focus on. And after that, she decided that she really wanted to focus on the tubules. She really thought it was amazing that they developed these vacuoles. And from her research, she was able to be comfortable or really understand that, yes, the proximal tubules, they're a hub of active transport for reabsorption. They have a lot of mitochondria, a lot of transport processes. really critical for kidney function, their target of injury and structural changes with diabetes and acute models of kidney injury and tubular interstitial injury. Can basically be a really good indicator of kidney disease, kidney function decline, and diseases like diabetes. So she was really interested in the proximal tubule. On top of that, it's a gap in knowledge. Basically from anything that's been done with regard to SV in the kidney, nobody has begun to look specifically at the different structures, at either protein expression or proteomic changes in proximal tubules. And so based on the preliminary data we had in the lab already on looking at abundance of glutathione, glutamyl-cysteine synthetase in whole kidneys, her hypothesis was that proximal tubules will exhibit proteomic changes in pathways that regulate redox homeostasis and stress response following exposure to SV. And so she proceeded by making new tissue sections and putting them on these special slides that, if you can see within here on the slide, there's a plastic membrane. So she laid the tissue sections right onto that plastic membrane. And then she basically went to the laser capture microdissection scope, and you can see she drew an outline around specific tubules. She cut them, and that's the before picture. This is after it's been cut out. And these are the tubules in the collection cap. And so she did that with kidney sections from all the different mice. She specifically used the proiodic acid shift stain to be able to see her samples with light microscopy and delineate the basement membranes so that she wasn't crossing from one tubule into another. From this, she processed those samples, she extracted the proteins, she worked with Danny Wilkie in Dr. Merchant's lab, and he did a fantastic job training her on, and another medical student from Dr. Merchant's lab. Both of them basically did a similar workflow. So they were able to extract the proteins and do their mass spectrometry magic on it. And they identified over 1,600 proteins from these formalin fixed paraffin embedded tubules. And after that, she started doing her three-way ANOVA analysis. And this is where then we actually, with the guidance and help of Dr. Adam Gaveda in our division, he helped. She organized all the data, forwarded it to him, and he was able to run some statistical analysis for her. And then she was able to crunch the data and begin to present it in a fashion for us to really understand. So we did a three-way analysis first because we had the three variables. We had sex, diet, and gavage, sodium benzoate or water. And it's pretty stringent. And even with that, when we look at that, you can definitely see when you take the main effects of SV, diet, or sex, the biggest factor determinant when you lump everything in, both male and females, was sex. So 588 unique proteins. There were 2 proteins common to all 3 from those 1,600. And so this was obvious then that in this three-way ANOVA, with sex being such a heavy determinant, that it's kind of masking what's happening in females or in males. She also looked, in addition to the SV alone effects, to the interactions, since this was in an ANOVA. So looking at either the sodium benzoate and diet interactions, sodium benzoate and sex, or sodium benzoate, sex, and diet interactions. Interestingly enough, this protein came up in both analyses. So in both the direct effects and in the interactions, and common to all 3. So she moved on, or Adam moved on to do a 2-way ANOVA analysis for us. So we did a 2-way ANOVA to look at the effects of sodium benzoate and high-fat diet in just females, and then doing the same thing in just males. And this was great because now we revealed unique proteins. So the females, looking at just the effects of sodium benzoate, they had 22 proteins that were significantly regulated. And they did not overlap with the 50 proteins that were regulated in the males. So completely unique changes. The interesting thing about doing the heat map is that if you look at overall, so she did 3 mice per group, and this, each one of these, this is 3 control mice, 3 control diet mice on SV, 3 high-fat diet mice, and then 3 high-fat diet with SV. And you can see if you just kind of look at the columns, SV has an effect to increase. So blue is increase, red is decrease. Has this general switch effect. High-fat diet kind of in the opposite, and then SV again reverses the effect of high-fat diet, or vice versa. So we just really have this nice separation of the treatments happening with the significantly altered protein expression in both the females and the males. One thing that stood out was this protein ACE2, so angiotensin converting enzyme 2. It came up through multiple analyses. So in the two-way ANOVA in males, it came up as the #1 ranked interaction between diet and sex. In the three-way ANOVA, for the interaction of all three, it was #3. Three-way ANOVA, direct sex effect, it was number 35. Three-way ANOVA, looking at the interaction of diet and SV, it was number 30. And when we looked at the two-way ANOVA interaction, this is what we saw looking at the multivariate chart. And so this is where Dr. Guedo really helped us try and understand the data and the interactions. And so as you can see, here is the abundance of ACE2 in controlled diet water. SV reduced the abundance. High-fat diet alone also reduced the abundance, but the combination of high-fat diet and SV reversed that, okay? Now this difference here, if you actually look at the y-axis, it looks like the differences are not that different. But this is significant. When she did a volcano plot to observe the differences between high-fat diet and SV in males compared to control diet, it was it had the highest p-value, not the it's not the highest subregulated, but it had the highest p-value. And the reason for that was that every single replicate in each group, the values were so close to each other. And that's why we had this really significant p-value. But more significantly is the fact that it's ACE2. Well, so compared to ACE1, we're familiar with angiotensinogen and its conversion by renin released from the kidney to angiotensin 1. And then angiotensin 2 being converted by ACE1 to angiotensin 2. Angiotensin 2 through binding on the angiotensin 1 receptor can lead to vasoconstriction, cell proliferation, hypertrophy, inflammation, fibrosis, atherosclerosis, and it's a target. So ACE1, both AT1R receptor blockers and ACE inhibitors are targets of therapy in kidney disease. Conversely, ACE2 takes angiotensin 2 and converts it to angiotensin 1,7, which through binding on the MAS receptor will actually have the opposite effect of angiotensin 2 acting through the ACE1 receptor. And so here we have a target that's actually or a protein that's actually a target potentially of therapy in kidney disease because it actually attenuates the effects of ACE1. And we see it being reduced by both sodium benzoate and high-fat diet. As a matter of fact, there's a really nice review fairly recently looking at, targeting ACE2 for treatment strategies in diabetic retinopathy. And more to come on this. Potentially this is going to be an area that's going to be explored more. But we were just really psyched to actually find something so relevant to kidney disease treatment coming up as such a strongly regulated protein and interactor in our proteomic analysis. Okay, so another thing she did that Olivia did in this summer was to actually look at modification of proteins by sodium benzoate. So sodium benzoate actually leads to lysine benzoylation. It's actually a modification that happens, and this modification is actually categorized under the group of lysine acylations that can occur. It's fairly recent, and definitely multiple lysine benzylation sites have been shown on histones, and they seem to be associated with sites of active transcription. This will also, in the process of this benzoyl group being added with, benzoyl CoA is basically the substrate to then add the benzoyl group to a protein or to a lysine. This will actually lead to the mass shift change in any respective peptide by 104 Daltons. So what she did was she took whole kidney extracts, okay, and she ran a Western blot. And so this is what this is. This is a Western blot with using that benzoyl lysine or anti-benzoyl lysine antibody. And so if you look at whole kidney extracts from normal mice, you don't see any reactivity. If you look at the control diet mice treated with SV, you see a lot of proteins being modified and reacting to that antibody. Again, the high-fat diet water, we don't see reactivity. We see it with the high-fat diet SV, same thing with the females. Okay, and so this is basically telling us proteins are getting modified. Okay, but this isn't whole kidney extracts. That's just her Ponzo S stain showing her relative protein loading for that specific Western blot. And so the next thing, oops, I skipped, sorry. The next thing she did was she actually wanted to see where is it happening in the kidney, because the Western blot was from whole kidney extract, and she wanted to make sure, is this modification happening in the proximal tubules? So she did an immunofluorescence stain, and you can see in the, pardon me, in the female mice on high-fat diet, the overall abundance of the benzoylated lysine is not changing. You can definitely see control diet with sodium benzoate. We see abundance of benzoylated lysine going up, and pretty much in every tubule type. It's not preferential of a proximal or distal tubule, and this is in the cortex. So you can, again, you can pretty much be guaranteed that 80% of them are proximal tubules here. Not really that much of a difference with high-fat diet and SV. Same thing with the males. You know, we on this, you know, this is preliminary. She did this summer. She's going to continue to do some more. We're not sure if this is just an artifact or if it's really something real happening. The diets do have a vitamin pack that could have compounds that can be converted to sodium benzoate. So it would not be unheard of that under those conditions, there could be a diet component leading to SB accumulation, but we didn't see it in the whole kidney extracts on the Western blot. But again, in the males with SV, we see increased abundance of benzoylated lysine in the kidneys. What's more importantly, though, it's happening in the nuclei. So this is really suggestive that potentially these are the histone modifications for which this modification, as it started being described within 5 to 10 years now, I mean, fairly new still, it's highly suggestive that those could be on histones. But that's something that we would have to explore, or she's going to have to explore as she continues with her research. So I was talking about the mass shift that would happen in proteins if they have that lysine modification. And so Mike Merchant and Danny Wilkie, they reanalyzed the proteomic data, trying to find peptides that had this mass shift related to benzoylated lysine. And what they found in all of the analysis of all the over 1,600 proteins that we analyzed, there were 2 proteins that came up to meet this requirement of this mass shift. And it was actually kind of interesting. One of them is EH domain containing protein 1. So this particular protein in individuals with proteinuria and a high frequency of hearing deficit, they have a homozygous missense variant. So in humans, there's proteinuria of low molecular weight proteins, and there's impairment of renal proximal tubule endocytosis of filtered proteins. And we saw a lot of proteins, you know, when she started to, she has begun to do her bioinformatic analysis, we keep having endocytic processes coming up. And so, you know, who knows? It would be really interesting to actually see whether or not modification of this protein could actually change albumin handling by the proximal tubules. Another protein that came up is protein tyrosine kinase 2 beta. Yes, sorry. This has a non-membrane spanning protein tyrosine kinase activity. It's involved in cell surface receptor signaling pathways and cell migration. It acts upstream of PI3 kinase and PKB signal, AKT signal transduction. And the human ortholog of this interacts with nephrocystin, which is primarily associated with nephronophthisis. Sorry, I have a hard time pronouncing this one. But it's a genetic kidney disorder. Again, just really interesting that the 2 proteins that were identified out of the 1,600 to potentially have this modification, they both have such a strong relation to some forms of kidney disease already. So we're excited. Olivia's really excited about this data, and I'm sure she's going to want to explore it a little bit further. Anyway, so in summary, The high fat diet, you know, we saw baseline characteristics happening similar to what we've seen in the literature. Increase in body weight in males, no difference incurred by the addition of SV on either body weight or kidney weight, relative kidney weight. With sodium benzoate and or alone or the combination with high fat diet, we're not seeing rampant injury to the kidney. Focal, it makes the case then, given what we've seen with our proteomic analysis, that there are changes happening. Ice on a high fat diet or SV. What I haven't shown here is that Olivia, she did a completely separate cohort of captured tubules, specifically looking at the vacuolated ones. And so she still has to analyze those data. But there's some pretty interesting changes already in her initial analyses. So we see proteomic analysis reveals differential regulation of protein abundances between males and females. And it really, again, backs up what we were seeing with that really simple paper that I showed you with regard to the gross changes, gross dissection changes that we can see in unmasked compared to whole kidney, and makes the case to continue to basically separate specific tissues and or spatially resolve them with additional microscopy and or decrease the number of samples that are actually collected and analyzed. Future directions, like I said, Olivia started this project this summer. She's continuing with research, going into the physician scientist development track. And so she's going to be gearing pretty heavily for bioinformatic pathways analyses of proteomic data, and then tissue validation level of the mass spectrometry findings. So lastly, I want to thank Dr. Jofflin for the collaboration and suggesting that we look into this area. And it was a great collaboration, has been a great collaboration. For the group within the nephrology that were specifically involved in this SV project, Dr. Michael Merchant, who basically has been a collaborator with both Dr. Jofflin as well as a co-mentor for Olivia, Dr. Adam Gaveda, with all of his help with the analyses, statistical analysis. Danny Wilkie for everything he did training Olivia and helping her with analysis of her samples. Obviously, Olivia for really spearheading the proteomics and spatial resolution of protein changes in these samples, as well as some undergraduate students who did some of the histologic findings and the Western blots that I showed you earlier. I'd like to thank my other collaborators in nephrology who, you know, all the papers that I showed with some of the methods that we've used to separate tissue, they've all been involved in. And that's it. Thank you very much.
Anybody have any questions? There you go.
I've never heard of the benzoated lysine before, so that's kind of interesting. I mean, I was curious, from your Western blot, there's either dozens of proteins that are modified or a couple proteins that are heavily modified. So do you know, does that antibody precipitate? And have you thought about doing like a chip-seq kind of thing?
Yeah, they're actually, so there's some really nice publications where they've actually published the benzoylome, where they've done that. They've actually aminoprecipitated. Dr. Goffin had done that with the liver samples as well. So she had done an IP with this antibody and mass spectrometry analysis, and she had, you know, a whole host of interesting proteins. So it's a neat modification. If we don't, we do want to see like within the groups, with the addition of either a high fat diet or between the male and female, certainly differences beyond that. Because given what we said at the proteomic level, with the column switch, right, with the protein abundance, we're wondering if the SV benzoylate or SV induced benzoylation is also regulated.
There is a question in the chat area. I can excuse me, question. Question is, have these findings for high-fat diet been replicated in ketogenic diets? Noted the high-fat diet had plus 40% carbs, which is dramatically higher than what's used in therapeutic carb restriction, with the classic ratio being 4:1:1, or a maximum of 10% carbs, ideally less than 20 grams of total carbs.
Yeah, so for this study specifically, no, but that would be really interesting actually to have that composition difference. By weight, it's 21%. I guess by kcal percent, it's 42%. But in combination with SB, no. Actually, this is the only study where sodium benzoate treatment has been combined with high-fat diet.
That's all from the chat. So. Anyone else in the? Oh, Dr. Merchant.
Is SP good?
No, you can actually, if you if you're really bored and want to Google, you can go find some people that will rant about how some people are sort of catastrophizing SB, and that they say benzoic acid is naturally occurring. It is being converted in your body to sodium benzoate naturally. If you were to assume the natural conversion and exposure and metabolism and excretion compared to consistently being on an ultra processed food diet, then I think it would be different. And especially given that, at least in these mice, what we're seeing, if those changes are occurring and we know that they're happening in transcriptionally active sites, then that could be questionable if you were to stay at a steady state level. We definitely saw in the early analysis from her, we are seeing a lot of changes in amino acid metabolism induced by SB.
So SB, PFASs, plastics, and all these things are in the way you store foods. And so if you leave them heated, it converts so you've taken diet, which is like every day you take in all these calories from different sources. And then if you were to throw the complexity of the container the food comes out of and all the leached plastics and PFASs and all these things, you're sort of, how many hits are you getting if you're
it's like a little mini explosion in a container.
Right.
It could be potentially.
So do we have to dissect each one of those individually?
You could, if you want to actually read the microplastics in the container. I think there is a paper where they actually went around. I think it was in Africa. They went to multiple locations where they were storing 2 liters of soft drinks, and they were trying to measure the different components and see how much degradation there is, and or potential, not just degradation, but potentially if free SB is being lost, then is it being converted to benzene? Because a lot of those soft drinks and or drinks, sweetened drinks, you have ascorbic acid, you do have metals. And so if you're in sunshine, heat, if you have that perfect storm, then you have a lot of conversions occurring and bleaching on top of that.
And we all know benzene's bad.
Benzene's bad.
So I just had a question. I know you mentioned the sex differences have been reported. What is thought to be the main reason males are more susceptible to like the high fat diet?
Well, the vacuoles to the so basically what's happening is they're retaining a lipid droplet, and that's due to their mitochondrial function. There's specific mitochondrial enzymes that are actually leading to that.
So maybe SB is fine if you're female.
I mean, SB alone causes vacuolization in the males. You haven't done oil redo to confirm whether those vacuoles are lipid droplets or not.
We have a faculty member that loves Diet Mountain Dew. Just have to say, the high in sodium benzoate.
So you took a day's worth of sodium benzoate, the maximum allowable amount, and gave us an oral gavage. What do you think would happen if that day's amount was spread out through the whole day and given to the diet?
Yeah, so I mean, I had a better chance. So would it linger more, or would it actually be more effectively metabolized? I think that would be something to consider. If you give a whopping dose, like is it immediately? I mean, obviously with what we're seeing, we do have modifications that are happening. So it is, it's present, but I think that would be, if we saw a difference in the modifications, then maybe that can give us a better idea of whether it, in smaller doses, it would linger more. But it would be more realistic, I think, if it was in the diet, rather than a gavage, an oral gavage.